Hi class, this is the IMViC test. The IMViC is an acronym that stands for 4 tests. The I is indole test, M is methyl red test, V is Voges-Proskauer test, and the C is citrate utilization. The I between the V and C is just to help pronounce the acronym. So in today's lab we'll be inoculating the BROS, some methyl red Voges-Proskauer BROS for some of these tests, and then there is this completely different media that we'll use for the citrate utilization. We'll do in another lab. Alright, so we'll continue on with this and introduce the two labs. Today's lab is prepping for the Voges-Proskauer test and the indole test. We're going to use the same media, the Voges-Proskauer media, methyl red Voges-Proskauer media for that. The two labs are going to be in your lab manual, page 59 and 61. I would encourage you to go to the reference page for this Voges-Proskauer test. Kathy Jackson, Voges-Proskauer test, principles, procedures, and results. So you can look at that online, and that'll give you some background information into it. So what I'm going to do today is just inoculate these tubes, and this is what they look like. And then when we do the Voges-Proskauer test, we'll take 1 milliliter of the grown culture, put it in another tube, and add some chemical reagents, A and B, and look for a color change. The indole test will add another reagent to the remaining tube, bacteria, that's called Kovacs reagent, and then that will be a color change to be indicating whether there's indole. And so you look at that, you can look at that reference as well. It's Tank Shawar, indole test, principles, procedures, and results. So check those references out online. There's other tests that we've been doing that you could look at as well for background information. So I'm going to take a loop, incinerate it, sterilize it, and I'm going to go into, this is number 1, and all I do is just get a little bit of bacteria. It doesn't take much. You can't hardly see it. And then I go in aseptically, rotate this, put it in here, then I wiggle it around to get the bacteria to come off, flame that, and then I flame the loop, and then that's it. So I'll do this for the rest of them.